Well, welcome everyone to today's webinar. So good to have all of you with us. This is Rick Import for Fisher Healthcare, and our session today is how to start a next-generation sequencing lab for reproductive health research. Our presenters are Alain Rico, Senior Manager with Global Market Development, Reproductive Health at Thermo Fisher Scientific in France. He's coming to us from Paris today. And also Dr. Matt Osentoski. He is the Senior Manager for Global NGS Analytical Validation Services with also Thermo Fisher Scientific, and he's here in the U.S. and based in Atlanta. We thank our sponsor today, Fisher Healthcare, for sponsoring today's event. And now to our presenters for today. Alain Rico is an engineer in biotechnology with a degree from the University of Marseille, France. He is a member of the French Army Institute for Tropical Medicine and has worked on vaccine development studies in the fight against malaria. In 1997, he joined PE Biosystems, a multinational biotechnology product development company based in the U.S., which is now part of Thermo Fisher Scientific. He leads global market development for reproductive health and clinical sequencing solutions and coordinates collaboration with key opinion leaders and development of innovative solutions. He generated data for the first NGS-based workflow for combined analysis of PGT-A, PGT-M, and mitochondrial DNA, and is the initiator of the genetic analysis portfolio for assisted reproductive technologies at Thermo Fisher Scientific. Dr. Matt Osentoski received his PhD in biology from the University of Miami in Florida prior to joining Applied Biosystems, which is now part of Thermo Fisher Scientific, in 2006. As a field application scientist, he helped support and develop the first next-generation sequencing platforms and then served as a leader for NGS application support teams in North America. After a transition to clinical support, he currently leads a team of project managers delivering Thermo Fisher Scientific analytical validation consulting services for the Ion Torrent platforms. And Alain will be leading us off, so let me get you all set up on your slides and give you control. And welcome to you, Alain.
Hello, I trust you can hear me now. And I'm going to use my laser pointer. Perfect.
We hear you, we see you. Welcome.
Thank you very much, Rick. Matt and I are delighted to talk before this audience about how to start an NGS, a next generation sequencing laboratory for reproductive health research. In the next 40 minutes or 45 minutes or so, we will discuss about research on reproductive health, and in particular, Preimplantation genetic testing. We're going to show you what are the technologies used to perform this kind of analysis, why you should bring Preimplantation genetic testing for antibodies in-house, and we will show you that this is not complicated to build your own lab and validate your process. I'm told I speak too fast, so I'm going to try to speak slowly. But first, a few words about Thermo Fisher Scientific on top of what Rick mentioned. Our role is generally behind the scenes, if I may say. We provide products and services to life science, research, clinical, and industrial labs. But with 7,000 scientists and engineers and like $1.5 billion invested in R&D every year, we can offer a luxury of solutions. We are a world leader in serving science with industry-leading scale and an unmatched depth of capabilities. Our mission at Thermo Fisher Scientific is to enable our customers, hopefully you, to make the world healthier, cleaner, and safer. It is a very powerful mission, and it really resonates in the reproductive health area when it comes to, for example, ensuring healthy offspring. Now, let's briefly discuss the different reproductive health research solutions and Preimplantation genetic testing in particular. So for those of you not familiar with this application, here's a brief reminder about the purpose of the Preimplantation genetic testing research and what kind of events are being analyzed. These are the different genetic variations that are of interest of studying in embryos, from the single base and the single nucleotide polymorphisms, or SNPs, to whole chromosome events like trisomies or monosomies, via some more complex events like translocations, for example. So there are different types of pre-implantation genetic testing to study these different types of genetic variations. Actually, there are three main types of pre-implantation genetic testing. The first one, PGTA, standing for Preimplantation genetic testing of aneuploidies. It's when you are analyzing chromosomal aneuploidies, like, for example, trisomy 16 or 22. It was previously referred to as PGS for Preimplantation genetic screening. A variant of PGTA is PGT-SR, analyzing the structural rearrangements like unbalanced translocation. PGT-SR was referred to as PGD, Preimplantation genetic diagnosis. And finally, the third main PGT type is PGT-M, when testing for the variants relevant to monogenic diseases. PGT-M was also previously referred to as PGT. Now, you may ask why people even perform PGT. Well, PGT allows for the analysis of aneuploidy in embryos in order to select the best embryo for a future transfer. So it's kind of prioritization tool, if you may say. I will not go into details about why PGTA is of interest, but just a few details. Well, just a few words. There is a correlation between the maternal age, actually the age of the eggs, aneuploidy, miscarriages, and the risk of aneuploidy when prenatally diagnosed. The higher the age of the mother, and again, the age of the eggs, the higher the prevalence of aneuploidy becomes. So when the risk for a 30-year-old woman to have a live birth with aneuploidy is estimated to be 0.2%, compared to the risk of a blastocyst embryo having an aneuploidy, which is like 30%, you see that there is a huge difference. So for a woman of, quote, advanced maternal age, the likelihood of aneuploid results in PGTA testing can increase up to 85%. Thus, it is highly likely that embryos tested during PGTA will be abnormal. So in a nutshell, PGTA is a way to reduce the time to live birth for certain women while reducing the number of transfers required. It's less stress, it's less money involved. It's been 40 years now since some of the first in vitro fertilization procedures were performed, and in that time, there have been many advances in associated testing like PGTA. Nowadays, the vast majority of PGTA is happening on trophectoderm biopsy samples, where around 5 cells are removed from the external layer of the embryo through an opening created with a laser. Now, some clinics use the blastocellic fluid inside the embryo as it is perceived less invasive, but still it requires a fine needle to puncture the zona pellucida. And more recently, thanks to the sensitivity of the sequencing-based solution, we can now do real non-invasive PGTA using the DNA released by the embryo into its culture medium. This is a very good method. It's very sensitive to external contamination though, but it allows for analysis of embryos in territories where it is absolutely forbidden to perform a biopsy on an embryo. It may be your case. Whatever you choose, it's optimal to use a PGT solution that will work on all the 3 types of a sampling method. Very briefly, here are the different technologies in use for running PGTA. The historical solution first, fluorescent in situ hybridization or FISH. Very briefly, FISH is based on fluorescent probes binding specificity to a few regions of the chromosomes. In this example, if 3 chromosomes of 21 are present in the cell, there will be 3 colored dots that will be detected. So the number of targets is relatively limited, is very much limited, and so is the resolution. So later came the microarrays used for a comparative analysis of a sample to a known reference. This method, called ACGH, was the first chromosome comprehensive analysis solution, meaning it allowed to analyze all autosomes and sexual chromosomes at once. Still, the workflow is quite long and the number of samples is limited, in particular because by definition you need a reference, so for every run, so that's a sample less to analyze. The next evolution is the SNP arrays, much more advanced, much more informative. The SNP arrays allow for interrogation of hundreds of thousands, if not millions, of polymorphic single bases for a much higher resolution. There is no need for a reference sample anymore. But still, the workflows, even though somehow more automated, are long, and the throughput still is limited. Throughput in terms of how many samples per run you can analyze. And finally, the next generation sequencing, NGS. This technology allows for running much more samples, is much more flexible, and is really future-proof in terms of what kind of application you can run on the same system. NGS is also called massively parallel sequencing because millions and millions of DNA strands are sequenced in parallel. And if you would sequence one strand at a time, it would, like, for example, on capillary electrophoresis, it would take forever. So NGS is a very simple workflow with throughput, speed, and cost effectiveness. There are a few NGS methodology, but one, for example, is the semiconductor sequencing. So the name comes from the basic principle of this sequencing, ion semiconductor sequencing. It's using a semiconductor chip, similar to the CMOS chip you use, let's say, for the camera on your smartphone. This chip is used to detect a flow of hydrogen ions that are released during the incorporation of nucleotide while copying a template. The chip is covered with millions of tiny wells. And then the template is converted to a library by fragmenting the genome and ligating specific nucleic acid adapters to the fragments. The fragments are then bound to tiny bits, clonally amplified to generate enough signal during the sequencing, and then flowed into the chip. And actually, at the end, you have one bit in one well. Next step, the nucleotides, A, C, G, T, are sequentially sent to the chip to build a complementary strand against the template. And as the bases are incorporated, they release hydrogen ions, which changes the pH of the solution in the well. So under each tiny well, we have a tiny pH meter, basically. These changes are sequentially captured by a sensor below the well, as I said, and then we build what we call the sequencing reads, essentially a string of letters that correspond to a specific template. During the analysis of the data, the reads will later be mapped to the reference genome sequence in order to detect different types of genetic variations, as we are going to see in the next slide. So the advantages of this semiconductor technology is really that there is no laser, there is no optic, You can sequence short or long fragments. And eventually it is fast, very easy to implement, and requires much less maintenance, which is important for a lab equipment if you want to start your own lab. So with semiconductor NGS platform and NGS platform in general, we can detect all types of genetic events, whether it implies a single base or a whole chromosome. It is not the case for all the technologies, methodologies. So, for example, FISH, we mentioned, is restricted to regions of chromosome used to impute then the number of copies of the whole chromosome. ACGH cannot detect a single base event. SNP arrays, they need to know in advance the bases of interest. NGS is really the more versatile solution. There is no a priori knowledge required. Basically, we sequence everything and we analyze later. And depending on which algorithm we are using for the analysis, we can give a result about a specific type of event. One way to perform PGT analysis by NGS is to use the approach of low-pass whole genome sequencing. So this is a very long slide title. I apologize. It is powerful yet very easy to operate. And I'm going to show you that it is also easy to bring in-house. As I just said, one of the main methods used for preimplantation genetic testing of aneuploidies these days is the low-pass whole genome sequencing on next-generation sequencing platform. This is the one used on semiconductor sequencing platform. It is also known as a shallow whole genome sequencing. But the low-pass whole genome sequencing is basically a whole genome sequencing on a diet. Much less reads required. Thus, we have a very low coverage, a few percent or even less. But despite that very low coverage, the method is very sensitive. So, for example, on the semiconductor sequencing platform, one need only 100,000, 150,000 reads to analyze regions of a few megabases. So nice resolution. The good thing is, since much less reads are required for a sample, you can analyze much more sample per run, sometimes up to 192 samples per run. This is a lot indeed. So PGTA by low-pass whole genome sequencing on semiconductor sequencing platform, as I said, is fast, very simple to operate, and there's a lot of automation and then a minimum hands-on time. Basically, there are two versions of the sequencing platform. Systems with each step downwards library building, each step performed on a dedicated instrument, and systems with integration of all the three steps. Here you can see the difference between the two versions. On the integrated semiconductor sequencing platform, the templating step, the preparation of the beads, the sequencing step itself, and the analysis also is included into a single instrument. So it's a lot of analysis turnaround time and hands-on time too. Yet it's very flexible. And PGTA by low-pass whole genome sequencing on semiconductor sequencing platform is as fast as 10 hours for 16 samples, 10 hours from the biopsy to the results. And as I mentioned before, you can run typically up to 192 samples at a time. It's good to know also that within a run, each sample is barcoded with a unique molecular code, 10-ish base pair, allowing to multiplex to pull samples together and avoiding sample mix-up during the analysis. You exactly know which sequence comes from which array. Simple yet powerful PGTA by low-pass whole genome sequencing on semiconductor sequencing platforms make it easy to bring PGTA in-house, make it easy to expand your research offering and perform up-to-date analysis. So here's why you should do so. There is a growing demand for preimplantation genetic testing. And by insourcing, you keep control. You keep control of cost, you keep control of delivery time, of accuracy of the results. And overall, running PGTA in-house offers a very competitive advantage. You show that you are offering the up-to-date service to your customer. From a business point of view, typically, a specialist from the provider of the PGTA solutions can help you to optimize the return on investment and support your growth by expanding further the testing menu and or validate your processes. Eventually, by incorporating PGTA in-house, you will achieve a winning solution to perform a reliable NGS-based vulnerability analysis in your own lab. Now, here are some information to help you build your own lab, your own NGS lab. The following information I gather is based on a recommendation from scientific societies like ESHRE, the European Society of Human Reproduction and Embryology, and also from real-life example of implantation of PGTA in small clinics. This deck is not intended to be comprehensive, but it aims to give you a good overview and a sense of how bringing PGTA in-house is indeed an easy process. As a forward, it is always recommended that the genetic analysis lab is separated from the lab when you manage the oocyte and where you do the embryo biopsy. It's always better to separate. Now, within the genetic lab itself, and because the workflow of PGTA or other reproductive health applications typically involve an amplification by PCR, you must separate spaces where the pre and the post amplification occur. And I mean separate physically, either using hoods or different assigned rooms for each step. You must do that to prevent contamination by PCR product between two PGTA analyses. Indeed, you all know that PCR is exponentially amplifying the original DNA in the sample by thousands, if not millions of times. It means that a sample with a limited amount of DNA can easily be contaminated by a previously amplified sample, either through aerosols or even physical transfers. But this is critical to avoid, of course, when you are running a PGTA, as you start with a few cells or even just microliters of culture medium. It is important also when you are running carrier screening. It is in a way easy to do it, right? It's easy. It's easy to separate the clean space where the molecular biology reaction will be set up and the dirty space, all between quotes, with the amplified material. For the workflow starting with limited amount of DNA, like, again, an embryo biopsy, you should ideally use a dedicated hood with a dedicated thermocycler, etc. If you use only one room for the full process, use a hood to separate the space and make sure it is positioned away from the post-amplification workspace. And it's always better to dedicate material for each step. Dedicate pipettes to each step, dedicate vortex, dedicate, maybe thermocycler to each step. And even if using a dedicated room for each pre-amplification and post-amplification, which is, by the way, the best option, use a hood and dedicated material anyway. As a general recommendation, the access of the room should be restricted to authorized personnel only. It is also better that the rooms for changing clothes and washing are separate from the NGS laboratory. Same separate office space for administrative work should be available outside of the NGS laboratory. The area for cleaning and sterilization of material, if present, should be separate from the NGS laboratory. I guess you got my point. The working environment also is important for your employees. So follow the local, national, or even pan-national, like us in Europe, occupational health and safety requirements to provide a safe working environment that minimizes the risk of distraction and of fatigue. Because on top of your employees' work-life quality, the goal is to avoid making a mistake during the analysis, pipetting or switching samples. Examples, you can adjust for the bench height to your personnel. You can use adjustable chairs. You can define the adequate workspace per person. You can use efficiently the space and surfaces. You can use sufficient environmental lighting and air conditioning with control humidity, with control temperature, etc. Also, think when you are designing the lab and when you are choosing maybe the material for construction, to make sure to minimize the release of volatile organic compound, VOC. And by the way, it's the same for the lab, for the embryo lab. Try to use things that will not impact, that will not be toxic toward the embryo. The clean space with the hood on the left, the dirty space on the right. And in between, you have the systems for templating and sequencing. You see freezer and fridge. This is a centrifuge. You have a spectrophotometer for quantitation, a thermocycler, obviously, vortex, pipettes, etc. You can put the analysis servers and the UPS under the bench. You can also put in the hood a small centrifuge and a vortex and a dedicated set of pipettes. And as I said, try to avoid contamination. And one way is if you are not using the full capacity of a run, let's say you only run 24 samples at a time, use the other barcodes available for the next run. Do not use again the same 24. It will help you to minimize the risk. Some labs may want to implement a witnessing system when the second operator monitors and checks the main operator does not make a mistake. And more, you switch sample tubes by mistake or invert samples before the barcoding happens. If you are using an integrated next generation system like the integrated semiconductor sequencer, which, as I explained earlier, combine everything required for templating, sequencing, and analysis into a single box, you can save some real estate as shown here. So I'm going to switch back from one to the other, and you can see that you can save some space with a single box to do everything. Here's a version, obviously, with 2 rooms, 12 by 12 feet. That's more or less 3.6 by 3.6 meters. You can have a door between the 2 rooms, but if you do so, do not go back from dirty to clean. And even if you have individual doors, once you left typically the dirty room, do not go back the same day, or at the very minimum, not coming back. Do not go back to the clean room with the same lab coat at the very minimum. And again, the same version of a 2-room lab design with the integrated semiconductor sequencing to save space. So I'm going to also switch back and again, just to show you that you can save some limit. All right. Now, I'm checking on time. Let me go into details for some of the elements we just discussed. The hood. It can be a basic pipetting hood, but at least you should have a UV light system because you need that to clean by breaking the DNA molecules. It's always better to use a hood with a laminar flow also because it will avoid that the contamination from outside will come into the hood. That said, once the barcodes are added to the samples, it's possible to work on the post-PCR area without a hood. But if you can have two hoods, it's always better. I mentioned it a few times already now. You need to make sure that you do not contaminate your samples or the molecular biology reactions with DNA material from outside. The DNA material from outside can come from an operator, a technician, either cells or fluids. It can also come from amplified material from previous. So it's critical, but yet there are simple rules to include in your own standard operation protocol that will help you to do so. So use clothing, including gloves and hair cover. It's even more critical for non-invasive PGTA when we start from very, very little amount of DNA release in the embryo culture media. Change glove after each step of the workflow. So for example, when you build the library, change glove after DNA extraction, change glove after the first linear pre-amplification, and after the amplification. All plastic wear, including tips, should be, well, DNA-free, that's kind of obvious, but also DNase and RNase free. A good option is always to use dedicated specific set of everything, dedicated set of pipettes, tips for the different steps, for library construction, for quantification, for instrument setup. Finally, you can use decontamination, DNA decontamination solution, like 1% bleach or UV irradiation, typically 244 nanometers for 5 minutes, or half an hour after that. Do that after each workflow step. And at the end of the run, typically you can wash everything again with sodium hydroxide, for example. In terms of ancillary equipment, an NGS lab, at the end of the day, is a basic molecular biology lab. So of course you need a thermocycler for the qPCR steps, and you need accurate pipettes with secure attachments so that the tips stay in place, don't go away, and the pipetted volumes are the good ones. You need vortex, you need plate centrifuges, you need tube centrifuges, small ones to go on the bench or in the hood. For the analysis, you will need a server. We recommend always a server. On-site analysis is always better compared to cloud analysis because, one, you keep control of your data, and two, it's typically much faster. You don't have to upload. So unless you are using an integrated sequencer, which again includes the analysis server, get a server dedicated to the analysis. And then depending on your policy and the regulation in your territory, you may also want a server to store the data. Finally, it's always safer to secure the power in your lab with a UPS. So again, a tip, put UPS and server under the bench if they need to be in the same room, otherwise put them in another room. Talking about benches, you only need to make sure that they can stand a few hundred pounds and they are steady, since some instruments, like centrifuges, can generate vibrations. So do not install an equipment generating vibrations on the same deck as an instrument sensitive, like using pipettes or sensor. What you can do also is you may consider to use an anti-vibration table if your lab is located on a very, very high floor in the building, or if you are near a transportation like a train station. Next, storage. So nowadays, kits for running PGTA come as ready-to-use cartridges or bottles, and small bottles with tubes, either stored frozen, cool, or at room temperature. So you will also need to store sometimes the libraries during the run if you need to reuse them or if it is in your SOP to do so. So in consequence, you need to plan for adequate storage space. So even though a fridge-freezer combo may be enough to start with, depending on how many times you run, you will need more space, so maybe a standalone dedicated freezer will be required. And the one tip I have for you is be careful. Sometimes the instruments you are using, they need access from the top for service, for maintenance. So if so, don't install shelves on top of this instrument. In terms of air management, using a positive pressure in the lab is always a good idea. All potential contaminants as aerosol will stay outside. And I mentioned before the need to avoid VOC. So make sure that if you use filters, to use high efficiency particulate air filters that also retain VOC. It's not necessarily the case, so check with your provider. I'm almost done, Matt. You have this great lab built and running, and you generate perfect PGT data by low-pass whole genome sequencing on semiconductor sequencer. Now, how to analyze and manage all this data? In terms of analysis, you don't need a bioinformatician. You should not need a bioinformatician. So make sure that you are using a PGT solution that use workflows, generating easy to interpret results as plots and tables, like you can see here in this example. Plots on the left here show ploidy on the y-axis and the different chromosome on x-axis, with or without smoothing, detection of segmental events, mosaicism, etc. And the same information is on the table on the right. And to manage the data flow, you may want to establish a laboratory information management system, LIMS. You may want to do that. The job of a LIMS is to give you insights about beyond what you would get from a simple sample tracking. It should help you to understand the health of your lab, ease the lives of your staff, and ensure the highest level of conformity to regulatory standards. LIMS can be used to support your lab also with a variety of connectivity endpoints, automation interfaces, and statistical methods. And as a LIMS solution, it should support you at any instrument or automation touchpoints. It should integrate seamlessly to automate the pooling of QC values, generation of liquid handler files, etc., etc. Finally, a LIMS should also support ancillary activities beyond NGS workflow, with best-in-class biorepository, for example. And it should also manage a full suite of analytical tools to automate your KPI tracking, etc. So think about the LIMS. It can really make your life much easier and your process stronger. Finally, we talk a lot about materials. So what about the humans required to run an NGS PGT research lab? I already mentioned that you don't need a bioinformatician, but really to run your lab, you need a single employee, a single good technician, typically owning a 2-year degree in molecular biology. You can always decide to use a higher profile if you are running a CAP-accredited lab. And we discussed the possibility to witness some steps, so you may want to adapt the number of employees accordingly. Continuous training is a must, and validation of the knowledge too. So ask your solution provider to help you with that. As far as interpreting the results and validating the results, what I call the desk, really the requirement will vary depending on the country. So typically, it's a director level with a PhD or a medical degree. And again, just for the bench, continuous training and validation is a must.
No, thank you, Alain. I really nice kind of setup, a very nice walkthrough of of how the process should work, a little bit about how the lab is set up, personnel, all those nice kind of upfront things that you need in place to get running in your own lab. The next part we really want to cover here is quality and competence, right? Every lab is going to need a QMS or a quality management system in place, right? And this could be to different regulatory bodies depending on what your desired accreditation is. But essentially, what is that going to do? It's going to govern the entire lab, right? SOPs in place for things like sample handling, for storage, data storage, checking your refrigerators and freezers. All of those things need to be in place and documented, right? So they can be audited at a later time. Things like calibration and preventive maintenance of pipetters, centrifuge, all the PCR machines, everything that you're using, right? It's important to make sure that they're performing the way they should be and to document them as well. Your personnel being trained, right? Having, again, some level of competency in place, whether that's through just for training or whether that's competency assessment from a regulatory organization that may actually send unknown samples to demonstrate your proficiency. But all of that must kind of be in place. And again, that's going to vary by organization to organization. How is your instrument running, both from a qualification standpoint and a qualification of the performance of the entire workflow across that, right? All of these are important to kind of demonstrate that your lab is able to produce these results over and over again. And although there can be quite a few of them, it is better to kind of dig into the regulations, and I'll give you a little bit of an example here in a minute. What I'd like to focus on for much of the rest of the talk is the assay validation part here, which is, of course, what I'm work on as well. We focus primarily on analytical validation. Analytical validation, when do you really, it's a process where you're really documenting how the workflow performs, right? And how are you able to demonstrate with real data in your lab that the assay that you're using is performing the way you expect it to, right? Reproducibly, as well as a variety of other measures, which we'll go into a little bit more detail. So it's really providing that documentation to the regulatory bodies that you're actually, you're doing what you said you can do. I think that's important, right? Obviously, you want to be able to produce an accurate result for your customers. When do you do an AV? Well, anytime you add a new test, if you have a new test to your menu, you're going to need to do a full validation to demonstrate its performance. If you have a major change to the workflow, typically that involves some sort of major change to instrumentation or chemistry. But it can also be if you've added different targets or changed what your assay actually is, right? So in oncology, this might be you add markers or remove markers, that kind of thing. But if you want to expand how your test works, you'd have to revalidate the entire workflow for that to happen. Again, and I'll kind of hopefully walk you through what needs to be done for that. For the most case, instruments need to be, most of the regulatory bodies insist that that each instrument be fully validated. So there's complete interchangeability between those two. But again, you need to check with which bodies you're working with or what you want to get your accreditation from. So I want to kind of give a little bit higher level view here of an example for PGTA. There are different regulatory bodies here involved, one being, CAP, or the College of Anatomical Pathologists, things like New York State Department of Health, et cetera. And what I did in this slide is really summarize what are the high level tests that you need to do for a PGTA. And this is, again, there's more details when you dig in, but these are the basic ones, right? And the reason I wanted to show this slide really is to show, if you read across from right to left, many of the tests are the same, right? Some of them have slightly different names. Some have some expanded, specificity or expanded testing that might be needed. Than others, but ultimately it comes down to a group of tests that are all very, very similar in nature. So, so it's easy to get intimidated, but when you look into these regulations more clearly, there's a lot of commonalities in them, right? Depending again on what you want to do. I wanted to spend a little bit of time taking you through an example, and I'm going to take you through an example for CAP. Again, this is very high level, but one of the things I wanted to point out, right, is, for many of these regulatory bodies, you have checklists or documents that you use to guide you, and I've actually laid that out here for CAP. This is the molecular checklist, and I'm showing the first test we would be doing is accuracy. Accuracy is obviously important, right? This is evaluating characterized samples to really show that the test can correctly call an aneuploidy, right? So this is really going to involve the use of different commercial controls, which could be cell lines, but also you're going to want to include some embryo biopsies, right? Typically 20 or more, depending how many you can get your hands on. And you want to have samples that are both have aneuploidies and some that do not, right? But it's important to run these through and get as most varied number of mutation types or aneuploidy types as possible to make sure that the test is covering the range of different aneuploidies that you hope to detect within the assay. Again, it's important to include negative ones in there, but it's also super important to include embryo biopsies, right? Because that's going to be your input on a regular basis for this test, right? And Alana alluded to that a little bit up front. So analytical accuracy is certainly one a very high important one here. We're also sometimes we'll be adding an accuracy based on the base calling and the way the actual instrument is performing from an accuracy standpoint. If you're calling SNPs or things like that. In this case, we're going to be relying primarily on it calling aneuploidies. The next one down here is analytical sensitivity. Oftentimes when you're dealing with oncology testing, which is one of the things we do quite a bit of as well, is going to be limited detection. Well, obviously, in this case, we're not dealing with the tumor, we're dealing with the embryo. So the important feature we want to try to address here in terms of the sensitivity of calling aneuploidies is mosaicism, right? Or contamination across, right? So we want to make sure that the assay can work in the presence of non-aneuploidy DNA. So can it actually do that? So to accomplish this, we're going to do a controlled experiment where we're going to use a set of dilutions of normal and aneuploid cell lines at different percentages to ensure that the test can actually detect them there across that amount. And that can vary, and it's certainly something, I gave an example here, 40, 50, 60%, and that can kind of change along the line. Analytical specificity. In this case, the specificity is in terms of cross-reactivity with other genetic events or genetic variants, that may be present in the sample. So this is where it's important really to make sure that you're using some normal cell lines that are non-aneuploidy affected, just to make sure you're not calling any false positives, right? In that sense. So making sure the test is robust to that type of sample. Precision, having the same sample repeated within the same run. So in this case, they would take the biopsy and perform the workflow in triplicate. So this is 3 independent libraries that would be built, and those 3 libraries would be run on the same instrument. And this would ensure that you're producing the same run or the same result for the same sample for multiple preps on the same run, right? So obviously you should be robust to that, but it also is assessing that workflow up front and that library build that you need to do to load onto the instrument. Last but not least here in terms of the test is reproducibility. So instead of within a run, this is testing the same result across multiple operators and multiple runs using the same sample. So you'd pick a series of different operators if available, and then a variety of different samples. And again, walk them through the workflow independently, but in this case, you're going to run them on different runs run by different operators, right? So this is kind of ensuring that your technologist and your instrumentation are not having any effect on the results, right? And that you're able to produce a robust result in that sense. So, and again here, this is where I kind of comment again, most regulations require that each instrument needs to be validated for each assay that you're going to deliver on it. So you can see, I mean, the tests are not too crazy when you break them down in terms of what you would need from a high level. And then it's really thinking about how to actually run those tests and build the report that you would need out of the other side. So, I hope, I'll just kind of wrap it up here. I hope we've kind of demonstrated, that bringing PGTA, or any of these assays into your lab is not that complicated, right? It's getting things set up. It's very doable. There are a variety of different regulatory bodies and different scientific societies which you can kind of build your lab to those specifications to kind of get what you need for that. We have a variety of specialty support internally, not only from a set of user guides as well as FAQs, often online, but we have a very skilled team of specialists from our application side that are there to help you get up and running, train you initially, as well as help you if you had any stumbles within the workflow and optimization. Validation consulting services, that's what I do. Obviously, I'm very proud of what we're able to do and help people get up running, both quickly and efficiently. That can help save you time and ultimately save you money to go forward and get done your validations within your lab. So I hope you've learned a bit in this talk, and I will turn it over to Rick.
So a big thank you to both of you, Alain and Matt. Wonderful job, great presentations. Well, big thanks to our friends at Fisher Healthcare for sponsoring today's event. Huge thanks to all of you for joining us. We hope you'll join us again very soon for another Fisher Healthcare sponsored event in the very near future.